Hello everyone. In this video, we're going to be talking about enzymes and why they are so special and important for metabolic reactions. First of all, enzymes speed up metabolic reactions by lowering energy barriers that are needed for chemical reactions to take place. So, spontaneous reactions do not need any added energy, but they can be very slow, enough to be imperceptible. For example, the hydrolysis, the breakdown of sucrose into glucose and fructose, is spontaneous. It can happen. Um, but at room temperature, a solution of sucrose in sterile water could sit for years without um, appreciable hydrolysis, meaning they could sit for years without breaking down. That's not always good. Now, in a for biological molecules and for our metabolism, that's not good. So, um, chemical reactions in the body that only function spontaneously, you need a catalyst. It's a chemical agent that speeds up a reaction without being consumed by the reaction. And so enzymes are biological catalysts. It's remember enzymes are macromolecules, usually proteins that act as a catalyst to speed up the specific reactions. For example, adding the enzyme sucrase to the sucrose solution at room temperature will catalyze the complete hydrolysis of sucrose within seconds. It will literally help digest it within seconds. And so this is just showing you that sucrose plus water with sucrase being the enzyme, it will help create the breakdown of the sucrose, creating glucose and fructose. So let's talk about the activation energy barrier. Every chemical reaction between molecules involves bond breaking and bond forming. So a molecule must be contorted into a highly unstable state before the bonds can break to start the reaction. To reach this state, the molecule must absorb energy from its surroundings and enough energy to cause the breakage to occur. The initial energy needed to break the bonds of reactants is called the activation energy. Heat in the form of thermal energy absorbed from the surroundings often supplies activation energy. So once something absorbs enough heat, it's able to have now enough energy to break the bonds. And so the molecules become unstable when enough energy is absorbed to break those bonds. This is called a transition state. As atoms settle into new, more stable bonds, energy is released back into the environment, into the surroundings. So, in an exergonic reaction, the formation of new bonds releases energy that was invested in breaking the old bonds. So, this is showing you the progress of of a reaction. So, you have reactants here that will react together after they have enough activation energy, and then they will create products. And in this exergonic reaction, they have released energy. The products are now contain less free energy than the reactants. The activation energy provides a barrier that determines the rate of spontaneous reactions because not everything is able to absorb heat in a way to break or to gain enough energy to overcome this hump. It, not all reactants are able to get enough energy to transition towards products. For some reactions, activation energy is low enough that thermal energy at room temperature is sufficient to overcome the activation barrier, but most reactions have such a high activation energy that they need additional energy to reach that transitional state. Unfortunately, adding heat is not always a useful way to speed up reactions in cells because it can cause your proteins to denature. You can only get so hot. Heat is also impractical because it will speed up all reactions, not just those that are needed. Um, the amount of energy that is needed for all of your chemical reactions to occur in your body is beyond what is capable of biological control. So, if you think about it, your bodies are 98.6 degrees. That's pretty hot. If you think about it, you're pretty hot as an organism. And it's because of all the energy that's being released from your chemical reactions. Um, it is so important to control the amount of heat that there's even a whole entire part of your brain that is just um, programmed to control and regulate your temperature. So, increasing your temperature is not a viable solution. So, instead, organisms carry out um, uh, catalysts, the process by which uh, a catalyst selectively speeds up a reaction without itself being consumed. So, an enzyme catalyzes a reaction by lowering the amount of activation energy needed for the chemical reaction to occur. So, an enzyme cannot change the free energy, it only speeds up a reaction that would eventually occur anyway. And usually, the one way that it speeds up a reaction is that enzymes will find the reactants, bring them together so that they can chemically react. A lot of energy is spent trying to bring reactants together so that they can react. So in this diagram, it shows that in the black line, it is the normal um, activation energy that would be needed without an enzyme, and that with an enzyme, it now lowers the amount of activation energy it is to do the chemical reaction. Um, so enzymes are unique in that they're catalysts. Another specialty of enzymes is that they're very specific. They have substrate specificity. So enzymes um, are designed to only interact with one specific substrate. The reactant that an enzyme acts on is called its substrate. The enzyme will bind to the substrate and form what it calls an enzyme substrate complex. While bound together, the catalytic activity of the enzyme converts the substrate into a product. So most enzymes end in the word ACE. So if you see something that ends in the word ACE, it's generally an enzyme. For example, the enzyme sucrase is what catalyzes um, sucrose. It causes sucrose to break down into glucose and fructose. Sucrase will only bind to sucrose and it will only break down sucrose into glucose and fructose. It will not do anything else. It will never do anything else. It's that specific. Each enzyme catalyzes a specific reaction and can recognize its specific substrate among evenly closely related compounds, even among isomers, it will only do and react with the one substrate. Now, the region of the enzyme where substrates can attach to is called the active site. The active site. It's often a pocket or groove that binds to the substrate, and it has what we call complementary fit between the shape of the active site and the shape of the substrate, and that they will like go hand in hand. So if you put your hand in hand, that's kind of like how enzymes work. When the substrate enters the active site, the enzyme will change shape slightly, tightening around the substrate like a handshake. And this is called an induced fit, which results from the interaction of the substrate and the active site. It brings the chemical groups of the active site into positions that enhance catalysis of the reaction. So this is showing you an enzyme, which is located in blue here, and you have a substrate. That substrate will bind to the active site. And once it binds to the active site, you can see here, it's created an induced fit. In other words, the enzyme has fitted or closed around that substrate, creating an enzyme substrate complex. Now, the substrate is typically held in the enzyme's active site by weak bonds, such as hydrogen bonds, and then the conversion of the substrate to a product happens rapidly, producing products that's released from the active site. Um, enzymes will emerge from their active site without a change. And so let's go over the process of catalytic process of enzymes with a substrate. So in number one here, you have substrates that will enter the active site of an enzyme. And as they enter the active site of the enzyme and an induced fit has occurred, the active site lowers the amount of energy that is needed to undergo these chemical reactions. A chemical reaction will occur, and so now products will be created, and so those products will then be released into the environment, and then the enzyme will return back to its original form, and so now it's able to then once again um, act or react with other substrates. And so this enzyme will keep working. This cycle will keep going um, indefinitely until there's no more substrate or until something comes along and tells the enzyme you need to stop working. So enzymes use a variety of mechanisms to lower the activation energy. Substrates may be oriented to facilitate the reaction. Substrates may be stretched to make the bonds easier to break. The active site may provide a microenvironment that favors the reaction. Amino acids in the active site may participate in the reaction. Enzymes will do what they are designed to do to speed up and act as catalysts for these reactions. If the enzyme is saturated, meaning that it has enough substrates in the enzyme, the reaction can only be sped up by adding more enzyme uh, to the um, to the area. Now, enzymes are not perfect. Enzyme activity can be affected by the environment, such as temperature and pH. So the local conditions can affect enzyme activity. It can also be affected by chemicals that specifically influence the enzyme. So temperature and pH are generally the example of things that affect enzymes. These are called enzyme optima conditions. Each enzyme has an optimal temperature and optimal pH at which it will reach its maximum possible reaction rate. Up to that point, the reaction rate will increase um, with increasing temperature. Beyond that point, the rate of reaction will begin to drop. Enzymes begin to denature at temperatures beyond their optimum. So the optimum temperature of for an enzyme is really dependent upon the enzyme, dependent upon the environment that it's adjusted to. Um, so, for example, in an organism, the different parts of the bodies can have different optima, but in different organisms can have different optima. For example, the optimum temperature for human enzymes is around 37 degrees Celsius, whereas the optimal temperature for thermophilic bacteria is about 75 degrees Celsius, right? If we were humans, if we were in this hot springs, it would be too hot for us. It would actually could kill us by being in contact with that. So the optimal temperatures for different enzymes. A human enzyme, for example, would be around 37 degrees, where the optimal temperature for that heat loving bacteria would be more around 75 degrees. And you can see the curves here for each enzyme is different because they are from um, adjusted to different environments. Each enzyme has an optimal pH, which is dependent on the environment, which is typically active. For example, the optimal pH for pepsin, which is a stomach enzyme, is 2, whereas the optimal pH for trypsin, which is an intestinal enzyme, is 8 or basic. So the optimal pH for these two enzymes in the same organism is very different from each other. Enzymes also sometimes need or have cofactors, which are non-protein helpers that bind to the enzyme permanently or reversibly with the substrate. Inorganic cofactors include metal atoms such as zinc, iron, and copper in ionic form. Organic cofactors are also called coenzymes. You'll see that word used quite often. And so this is why vitamins are very important. Most vitamins either act as coenzymes or provide the raw materials to make them. I suggest for you guys to do vitamin D enzymes and magnesium. Take those together. That's actually um, I know helping a lot of people. Now we've been talking about ways that enzymes work, how to get enzymes to work um, optimally. But what if we need to inhibit enzymes? Or what if there's things that do inhibit enzymes? So there are certain chemicals that selectively inhibit the action of specific enzymes. If an inhibitor forms a covalent bond with the enzyme, then the inhibition is usually irreversible. That enzyme is now no longer functional. Many inhibitors bind to the enzyme by weak interactions, resulting in reversible inhibition. So there are competitive inhibitors that closely resemble the substrate and can bind to the enzyme's active site. Um, the enzyme productivity is reduced because the inhibitor blocks the substrate from entering the active site. Uh, Increasing substrate concentration can overcome this type of inhibition. And so this is showing you a normal binding where the substrate will bind to the active site of the enzyme, but then you can have a competitor come along and inhibit and prevent the substrate from binding, stopping the enzyme activity. There's non-competitive inhibitors that bind to other parts of the enzyme that causes the active site to change shape and makes the active site less effective. So non-competitor inhibitors can bind to a different part of the enzyme, changing the shape of the active site, and the substrate won't be able to bind to the enzyme like it normally would. Now, we know that there are toxins and poisons that are often irreversible enzyme inhibitors. For example, sarin gas was used in a chemical attack in Syria in 2017, killing and injuring hundreds of people. Sarin will bind covalently to the active site of acetylcholine esterase, which is an enzyme important in the nervous system. Um, this irreversible enzyme is what caused this irreversible competitor to that blocked the enzyme activity is what caused the people to die. Other examples of irreversible inhibitors are pesticides and antibiotics. So we have designed pesticides that are used to attack the enzymes, for example, in insects to prevent them from basically killing the insect, preventing them from eating our crops. And then, of course, we can, or your cells, your bodies create antibiotics to create antigens, or we can create medically antibiotics that are used to kill usually bacterial cells. So, regulation of enzyme activity can help control your metabolism. Um, Sometimes you need enzymes to work more efficiently. Sometimes you need enzymes to just stop working at all. So, chemical chaos would result if a cell's metabolic pathways were operating simultaneously. If everything was doing everything at one time, it would be chaos. So cells regulate metabolic pathways by switching on or off genes that encode for specific enzymes, or by regulating the activity of enzymes that already exist. One of the ways to regulate enzymes is through allosteric regulation. It occurs when you create a regulatory molecule that binds to a protein at one site and affects the protein's function at another. This type of regulation may either inhibit or stimulate activity. So most allosteric regulated enzymes are made from polypeptide subunits, each with its own active site. Um, this causes the subunits, the enzymes, to oscillate between two different shapes, one that's active, one that's inactive. Uh, So let's just show you. So in this diagram, it's showing you allosteric activators, inhibitors. So for example, in the top form here, you have four active sites and you have a regulatory site. And so it can oscillate between a functioning form and an inactive form, uh, depending on if it has an activator or an inhibitor. If you have an activator that binds to it, it will allow for substrates to bind to the active sites and it can do its job. But you can add an inhibitor that will cause it to change shape, where substrates cannot bind to the active site and it just stops working. It doesn't mean that the enzyme is damaged or destroyed. It just means that it's taking a break. In cooperativity, substrate binding to one active site can trigger a shape change to the enzyme that stabilizes the active form for all other sites. This mechanism amplifies the response by priming the enzyme to act on additional substrate molecules more readily. So in this case, you have an inactive form of the enzyme. So in cooperativity, uh, if you add a substrate binds to it, now the it becomes active and all the active sites will become active. Another form, I think it's the last form of enzyme regulation, is feedback inhibition. In feedback inhibition, the end product of a metabolic pathway shuts down the pathway. In other words, once you've done your job, you're no longer needed, so you can just turn off and wait until you are needed. It prevents a cell from wasting chemical resources by synthesizing more product than is needed. So in this example, you have a substrate, and that substrate binds to the enzyme, and it will go through a series of chemical reactions, creating an end product. When you create that end product actually will bind back to the enzyme, changing the shape. The active site is no longer available, and so that enzyme has been turned off, and it's waiting. Once the product has been used up, then the active site, the enzyme will return back to normal and wait for the initial substrate again. It's just feedback inhibition. There's a lot of processes that happen in the body this way. So, localization of enzymes within the cells, there's compartmentalization of the cell. It helps to bring order to metabolic pathways. Remember, there are some places where enzymes are more located than others, like the endoplasmic reticulum. Um, Sometimes metabolic pathways evolve to have multi-enzyme complexes. And then remember, some enzymes act as structural components of particular membranes, like in the plasma membrane. In eukaryotic cells, some enzymes reside within specific organelles. For example, enzymes for the second and third stages of cellular respiration are located within the mitochondria, not in the cytosol. And this is just showing you a mitochondria and how the enzymes are located in different spots of the cell. And with that, that concludes chapter 8. So in chapter 8, it's all about the laws of thermodynamics, free energy, how ATP works, and then the coupling of exergonic and endergonic reactions, and how enzymes are able to do um, and act as catalysts for all of the chemical reactions in your cell.